Okay. My name is Shaohua Wang, and I'm from Department of Neurobiology and Behavior at SUNY at Stony Brook. And today I'm going to give you a brief introduction to a kind of new technique in neuroscience, which will make your science colorful. Okay. So first, what do you think of neuroscience? Do you think it is gloomy because we have to do experiments all day long? Or do you think it is bloody because every time we'll put or plug an electrode into the mouse or some animal's brain to just activate it? Now I'm going to tell you something super. That is, in this video, the scientist can trigger the mouse by turning on the light. The light can activate the mouse to run or walk in circle. This neat technique is really powerful to help us to study the mechanism of how of the animal behavior, like how can we walk, how can we control our behavior and method. This technique is called optogenetics. Today I'm going to give you some basic ideas about optogenetics, like what is optogenetics, how does it work, and give you some examples of using it in the study. First, let's see what is optogenetics. From the part, I'm not going to give you a long and boring definition of it, but let's see some cool thing. From the word optogenetics, we can say that optogenetics can be divided into two parts. The first is opto, the other is genetics. Let's see opto first. All the stories start with this kind of green bacteria called algae. Algae is a strange bacteria that when there's light, it will run away from the light. And scientists found that it is because a kind of molecule on the cell membrane of the algae that makes it driven away, called channel rhodopsin. When there's blue light, the conformation of this channel will change, and there will be some cations like sodium goes through the channel and into the cell, and this will trigger the fire of the cell and makes the algae run off of the light. And so it is very similar to the process in mammalians' neural behavior. So neuroscientists want to find a way to make this molecule help our research in neuroscience. So they use some genetic ways. First, we get a gene of this kind of channel called channel rhodopsin from the algae, and we add some promoter, which is the switch to activate the gene together, and we transfer this gene into a virus and inject this virus into the mouse brain, and the promoter can help the channel rhodopsin gene expressed onto the membrane of the mouse nerve cells. So we can finally get channel rhodopsin into a nerve cell of mouse. After that, we'll see how it works. After we transgenetic the channel rhodopsin molecule, like this, in the neuron of mouse, there will be the channel rhodopsin on the cell membrane of the neurons. After we give it a blue light to trigger it, there was some sodium or some other cations flow into the neurons and trigger the neurons to fire, like activate it. And this one will, just like what we did, use electrode before to control the behavior of mouse. So now let's see some examples of it. This one is in a fly. Use the optogenetics technique. We can see there's a when there's a light, the fly is going to flee or jump, and because there's a in a box, so it cannot fly, but just jump in the box. And here is some other things, except for just activate some cells. Some other kind of optogenetic channels can inhibit the cells. Here is an example called halorhodopsin. This is has the same mechanism of channel rhodopsin, but its effect is to inhibit the animal's behavior. Like in this, after scientists can transgenetic the halorhodopsin into the worm, when there's a yellow light, the movement of this worm will be inhibited. And the next one is yes, that's what we saw before from the first page. Scientists use blue light to control the mouse's running or motor behavior. And also it is used in Parkinson's disease research. When there's a light there, it will, as we all know, as you may know, the Parkinson's disease is kind of movement disorder. So when there's a light there, it can control the movement of, can control the cell to give a right movement. So just because the new optogenetics is shining everywhere from animal behavior research to the protein interaction into cells, New Nature Method Journal has selected this kind of method as the method of year in 2010. And now, what do you think of the neuroscience is if we use this kind of super technique into it? That's all what I'm going to talk about. Thank you. So, any questions? Okay.
Will this technique could control mind in the future?
Mind controlling is kind of a complicated word. So we can control the movement of a single cell or a population of cells, but not control the mind. I mean, in the near future. But I think we can, if we know the exact mechanism of it, we can control it in the future. But it will be a long way to go.
I have a question. You said you can use light to somewhat control the neural cells. The sodium concentration will change within and outside of the cell.
Yeah.
So in that way, you can do some. Can you do some prediction on how the animal behavior after light?
Yeah. Well, actually, what we can control from optogenetics is to mimic the neuron activity with this technique. So influx of sodium or other cations is it's just like the excitation process of normal cells. When the neurons are activated in physiological status, there will be cations influx into the neurons. That's the same way. But this one, what we do is to control this kind of influx, because we don't know, and in human body we don't know when the cations will go through the channels and into neurons. But in this way we can control that kind of flux. And if there's influx, there will be activation. Yeah. I clear? Yeah.
Is there any side effect, or is this method gonna harm your cell? Anything like that?
Yeah, well, actually, I'm not going talking too more about the safety or side effect. Actually, it is as safe as you can think, because it's not like the electrode to plug directly into your brain, which can cause some side effect. Because actually, when you stimulate the cells you want to stimulate, you also activate some other cells around it. But for this one, we can use some. You see here, there's a there's a promoter here. The promoter is the switch of the gene. So if we change the promoter, that we can control the gene to express in specific cells. So we can just, if there's light, we can just trigger the cell with this promoter. So we can control the cells we activated. That's it. Any more questions?
Good, thank you.